Okay, so thank you all for your interesting sort of, mini seminars. Um, Looks like some very interesting kind of leads in there that are obviously very relevant to the areas of research that dkNET serves. So I didn't have any specific kind of format for this particular session, but like kind of on a high level, what I thought I would do would be to give you first of all some insights into the work that has gone in, you know. Sort of on the back end, as it were. So all the work that has been done offline that allows us to basically present the information and the experimental information on the website, on the SPP website. So there's a lot of work that has gone into identifying the data and also annotating it in a way that makes it accessible to researchers. And so I think giving you some insight into that will sort of, I think, help you understand more the data science side of things. And then what I thought I would do then is maybe get some additional information from you as to what it is that you're looking for in the SPP, and those of you who are are planning to use it. I think that'll be a useful discussion for those of you who aren't as well, because it actually might actually give you some ideas that you might not have initially thought, because there are ways that you can use what's in SPP to infer regulatory relationships that you might not have actually seen when you looked at it. So I've got some notes that I took on your talks, but I'd also like to get some additional information from you guys on exactly what it is that you're hoping to get from SPP. And actually, what I will also add in is an interesting use case, an article that I just saw on my Twitter feed this morning that looks like it has a potential reference to the mechanism of action of SARS-CoV-2 in terms of its impact on the integrity of the lung epithelium. But okay, so what I'll start with, what you're looking at now is, I think you're kind of familiar with at this point, is the SPP UI, and there are various ways that you can interact with the data. When we were actually thinking about how to design the site, we wanted to keep things as simple as possible. So compared to other sites that there's a lot happening in the UI, ads, various other things. We wanted to sort of dispense with that and only have in the UI the information that's absolutely essential for the user to understand or to get out of it what they're looking for. And so for somebody who's kind of new to the site, we have a section where we list all of the data sets. So you can, as you know, recently we've been kind of focusing on annotating data sets that are relevant to coronavirus infection. But there's a lot of other information on there also, and basically we have organized this into 5 or 6 high-level categories. There are those that are relevant to the signaling, which I emphasized up last week, but there are also data sets that are relevant to models, and the model category is what these recent viral infection data sets fall into, because they're models, right? So giving the cells, you're infecting the cells, and on the other end, you're getting out a list of transcripts that are upregulated and transcripts that are downregulated. But what the model won't inform upon is how that actually happens. What happens is that between the virus landing on the surface of the cell and a transcriptional event in that cell. And so when we had our seminar a couple of weeks ago, I kind of I gave you some insights into how we can use existing SPP data, which is relevant to receptors, enzymes, transcription factors, how we can sort of intercalate those data points on the one hand with those on the viral infection on the other, and anywhere we can see intersections or overlap, we can infer that the infection is having an effect. We don't know what that effect is, but it's having some effect on the signaling through a particular receptor, enzyme, transcription factor. So this is one way in which you can access the data. The other is for if you're actually approaching the resource with a specific question in mind. So again, I went through a few of these last week, but I'll just briefly go through those again. So if you're interested in regulation of a specific gene or specific transcript, for example, a couple of your little brief summaries mentioned a couple of things that you were interested in. Pick one, EP3. So one thing that I would kind of point out initially is when you enter a lab, you'll hear things, various factors being referred to by the names by which they were originally identified. But it's also important because one of the main areas of the focus of this summer series is to allow data to be interoperable, right? So that's the I in interoperable. And so if you have a particular routine that is known by a bunch of different names, and you have a bunch of different labs using their own name, you can see that that's going to make interoperability fairly difficult. So it's always important to bear in mind and to use, when you're getting presentations, or you're writing a manuscript, or you're writing a funding application, is to always have that official approved symbol in there. So, for example, EP3. That's a name that's probably very familiar to you, but its actual approved symbol is PTGR3. Similarly, with CGRP, that's approved symbol is a CA. And initially, when these symbols were kind of introduced, which was back kind of in the late 1990s, when I was in the lab, everyone was kind of wondering, why have they undertaken this effort to come up with this whole set of new names that, that's just one more name for us to remember. But that system, without that in place, efforts like SPP and others, it wouldn't be possible, because there is no consistent identifier by which these factors can be identified. So that's something that I would caution you to bear in mind. So if you enter the name of your particular gene of interest, what you'll get back at the back end is evidence, and that's all it is. It's evidence that your particular gene of interest is transcriptionally regulated by various nodes, by the members of various node families. And what you'll notice is that some nodes are more experimental data points for some nodes than for others. And the reason for that is actually partly historical, because when we were first launched, our resource was actually specific for nuclear receptors. And so our annotation pipeline was set up for a nuclear receptor. So you'll often see that there are more data points for nuclear receptors than for other things. But that also, to a certain extent, actually reflects what is actually archived, in the public archives, and that's particularly the case for things like estrogen receptor and androgen receptor. Does anybody have any idea why those particular receptors might be particularly well represented in the archives?
You could unmute yourself or type in the chat. Yeah.
Yeah, anybody? Okay, don't be shy.
I didn't see from group chat.
All right, so as you're probably aware, there's a very substantial amount of research money has been spent on two diseases in particular, namely breast cancer and prostate cancer. And two of the factors are two of the nodes that have been, exhaustively researched within the context of those diseases are androgen receptor in the context of prostate cancer, and estrogen receptor in the context of breast cancer. And so what has happened, and this is partly as a result of literature. One of the problems with the research literature is that it tends to focus very heavily on a number of well-defined functional systems. So there's a lot of research literature already on androgen receptor in prostate cancer, and there's a lot of existing literature on estrogen receptor in breast cancer, and the result is that those particular paradigms, which are undoubtedly important in the context of those diseases, what happens is they just become more and more exhaustively focused on. And so one of the things that we want to do with SPP is to use archived omics experiments, which by their nature have got much less of a particular focus on one particular regulatory system or regulatory paradigm, because they look over the entire transcriptome, and so they allow researchers to identify relationships that might not be readily noticeable from just looking at the research of literature. So now, as far as what actually sits underneath this user interface is we have on the back end, we have our own application that's visible only for us at SPP, and basically we use this what we call DMC to basically organize all of the metadata. So you have the individual experimental data points, right? So a full change of 3.4, a p-value of 1 to e to the minus, whatever it is, e to the minus 4. Those are the data for a particular transcript, but then around that particular piece of information, around that particular data point, there's a lot of contextual information. So metadata, and that helps the user understand the context and allows them to basically use that data point more easily to formulate research hypothesis. And so I'll just walk you through some of the interfaces that we use to organize our metadata. One of the most important pieces of information is the particular cell line, or the particular, in the case of animal models, is the organ or a tissue in which an experiment is done. Because what happens in one particular cell type or tissue or organ, it doesn't necessarily happen in others. And in the most extreme examples, you might have a particular transcript that in one organ or in one tissue or cell type is upregulated by a particular node, by a particular transcription factor, but in another, that same transcript, that same node, that node might actually function to repress expression of that transcript. And this is one of the ways in which the body is able to basically coordinate the functions of different organs, because you might want to have one particular process upregulated in one organ, but in another you might want to have that particular process switched off. And so you can exert that effect, body exert that effect by modulating the relationship between signaling nodes and the transcripts that they regulate. And so as you can see, we organize these what we call biosamples. So you have the kind of high level physiological system. You have then individual organs within that system. And then on the bottom level, you have the particular cell type. And what we don't have here, actually, but which we should have, and we hope to be integrating in the future, is basically mapping these cell lines to an ontology. And for those of you who know what that is, it's basically a fixed, it's a series of fixed, definitions, controlled vocabulary that basically identifies a particular cell line uniquely in this case. And so what we need to do at some point in the future is to map all these different cell lines up to that ontology, and that kind of brings us back to the idea of interoperability, because I'm not going to discuss it here, but all of this, everything that you see within SPP is available through our API, which allows for automated interactions with other resources. And so if we can use ontology terms wherever possible, those ontology terms are maintained by an independent third party, so that that makes it much easier for other resources to retrieve exactly what they need from our resource. So that's tissues and still lines. The other this screen that you're looking at right now is allows us to see, at a glance, each of the individual experiments and how those have been annotated. Now you'll see that recently, as I said, we've been focusing on the coronavirus data sets, but more typically, if I can find a suitable page here. Okay, you'll see that there are. I'll focus your attention here on a couple of columns, and the first one here is BSM, and basically what that stands for. If I can go to the BSM page, that stands for bioactive small molecule. So, and these are very important within SPP, because most of the experiments that we have in SPP involve experiments that involve a BSM, and in order to make those useful within our system, what we do is we basically map those to nodes. So recall that nodes refer to receptors, enzymes, transcription factors, and then conodes. And when we make that mapping, what we are saying is that any effect that particular small molecule has on the cell, in terms of regulation of transcription, because we know that it acts through a particular node, we can infer that node is responsible in part for those effects on transcription. And so we're not so much interested in these small molecules themselves as to what they interact with in the cell, and we can infer that at least a part of their function on a cell is through that particular node. Now, of course, they may be interacting with other things in the cell, like we don't know that. We can only go off what's known about a particular small molecule. So, in this case, you have a particular small molecule that is known as a specific inhibitor of SMAD3. However, for the purposes of our annotation, we prefer to use small alpha numeric symbols that are obviously unique for a particular small molecule. It's much easier for us to discuss a particular experiment, or to annotate if we're using these specific short symbols. And we came up with this idea really from what I was discussing earlier, which was the effort to come up with unique human and machine readable symbols that were specific for a particular gene. So we have this. We have a similar system in place now for SPP, and it works very well for us. One thing I would also point out is that BSMs can either be synthetic molecules, so synthetic small molecules that have been rationally designed to either inhibit or activate a particular node. In this case, this particular small molecule has been designed to inhibit the SMAD3 transcription factor. However, of course, other small molecules. Oops. Let me not use that one. Okay, yeah. Here's Indian hedgehog. So this particular small molecule is actually a natural peptide. It's encoded by a gene in the body, and it interacts with the patch receptor family. So we have synthetic compounds, which can be either experimental in nature, or they can be approved therapeutics, or we have small molecules that are actually encoded by a particular transcript. So that's BSMs. The other important system that we have is to basically classify the nodes. And when we were thinking about how to classify them, we were really benefiting from years, in some cases, of decades of work that had gone on before, where organizations have been set up with the purpose of classifying these particular categories of molecules. And one of the most important resources, for us at least, is the IUFAR, International Union of Pharmacology's Guide to Pharmacology, where they have basically classified lots of different types of receptors, and they've annotated these receptors in terms of the particular small molecules that they bind. And so we made a kind of a systematic effort to integrate this information into SPP, and we use not exclusively. We had to do some mappings on our own because they aren't in IUFAR, but for the most part we use IUFAR's mappings when we come across an experiment with a particular small molecule. We first look and see, okay, has it been mapped, or look at our resource, and if it's been mapped, then wonderful. If it hasn't, then we go back into the research literature and we find a particular article or study where it has been mapped to a specific node. So that's receptors. One of the other classifications that has actually been around since 1962 is the International Union of Biochemistry, and this was the first effort to really, impose some sort of order onto the classification of enzymes, and really this their work in basically organizing enzymes into 6 or 7 major sort of high level classes is what we used for SPP. Now, again, there have been some modifications because a lot of other enzymes have been discovered since then that actually don't fit into these categories. So we've had to modify them somewhat. But here's another example of where our work in kind of organizing omics data is really owes a large part of the ease with which we're able to do that is down to work that's been going on for the past 50 years now. And then finally, it's a more recent. Oops. Okay, I don't. My apologies. I thought I had this tab open. This is a much more recent effort to basically classify transcription factors. So again, we have used a 3-level classification that we use for all of the nodes, and that because we wanted to keep things as simple as possible on the UI. If you start going into multiple levels, you're going to impact the ease with which investigators can actually get at data. So we wanted to kind of strike a medium in between annotating things in as much detail as possible, but then on the front end, not letting that become an obstacle to people using the site. So we have just to go back to our nodes page here, we have this 3-level system here. So this not all these classifications had 3 levels. Some have more, and so we had to do a bit of work in order to make those classifications fit. So these aren't the SPP classification isn't an exact replica of those, but it works to serve our needs. And so if you note this 3-level classification here, this is how we organize the regulatory regulation reports on the front end. You have the category, and then the class. You have the family, and then you have the specific node. So when we met a couple of weeks ago, and we were doing those exercises, when I was getting feedback from you, I think 1 or 2 of you might have kind of misinterpreted what you're looking at here. So I just want to make sure that everyone understands what they're looking at here. So we have our particular gene of interest here, Reb1, and I'll pick a node at random here. So in this a particular experiment, this was a knockdown of a FOXA1 in LN cap cells, and that resulted in a 7-fold down regulation of a Reb1, which is pretty large. So what this particular individual experiment indicates is that loss of function of FOXA1 results in down regulation of a Reb1. So that would imply that within the cell, FOXA1 acts to increase expression of a Reb1. So it's a positive regulatory relationship. If you looked at that on its own, that would be one thing. But the way that we have organized the information here is that everything that's a like experiment you'll see on the same row. So this is another data point from a completely different experiment, a completely different article that again shows that the knockdown of a FOXA1 results in down regulation of a Reb1. So you can see here that what this interface allows you to do is to integrate different people, different pieces of evidence, sort of aggregate those into a single view. So if you see something in one experiment, you go, okay, that's interesting. But if you see the same thing in a completely different experiment from a completely different lab, that gives you more confidence that something's actually real. And in the more extreme cases where this is 17-beta estradiol, most of these experiments come from either a T47D or from MCF7 cells or from reproductive organs in mice. So there's multiple different experiments here that show that when you give these cells estradiol, you get upregulation of a Reb1. And this is the principle that's behind the consensome analysis that I talked about when we met a couple of weeks ago. And this allows us to rank transcripts or genes in response to a specific node. So the more often that a specific transcript is differentially expressed, when you hit a node with whatever it is, in this case, a small molecule, can be overexpression, of knockdown. The more often that a particular transcript responds to that, the higher it's ranked. And Reb1 is actually one of the most highest ranked transcripts within the estrogen receptor family of consensome. And this was known from the research literature. There's oodles of articles already out there. So this isn't exactly news, but what is news is that there are other things that are also really highly ranked within the ER consensome, for which, if you went into the research literature, you would not find that information. So it's out there. It's archived, but it's just not easily accessible. And easily is the important word, because if you don't make something easy to access, chances are that researchers won't have the expertise that they need to basically go in and find this information. So with that, hopefully that's given you some insight into the work that's kind of gone into organizing the data for SPP. What I wanted to sort of touch on next was the article that I saw, that I was referring to earlier, where they identified sphingosine 1 phosphate as a small molecule, native small molecule that was essential for lung repair. And this, because at the minute, everybody's focus is on SARS-CoV-2. This kind of got me thinking, because obviously one of the the effects of SARS-CoV-2 infection is damage to the lung epithelium. And while SPP at the minute doesn't have any information on metabolomics, so we don't have any metabolomics experiments in SPP. You can still use it to sort of get some picture as to what the effect of the nodes, or particular nodes within a cell, might be on a small molecule. And the way that you do that is you go in and you look at the biosynthetic pathways for small molecules, and in this case, sphingosine 1 phosphate. So the sphingosine 1 phosphate synthetic pathway basically originates with ceramide, and one of the enzymes that's important for the synthesis of sphingosine is ceramidase, and I can go to. One second here. So yeah, the enzyme that catalyzes the hydrolysis of the sphingolipid ceramides into sphingosine is ASAH1. And so if I run a search within SPP for ASAH1, what I will get back is, as we discussed earlier, information on nodes, but also models within which ASAH1 is actually regulated. What I will say is that we are looking right now at our internal website. We are hoping to actually push our new updates out onto the public website this afternoon. But right now we're looking at our internal development version of SPP. So we have a bunch of recent RNA-seq data sets that have looked at the impact of SARS-CoV-2 in expression within the lung epithelium, and hopefully this query will run. There we go. So if you look down here at the bottom here, these are all the data sets, the experiments that relate to infection by SARS-CoV-2. And you can see that basically all but one of these, ASAH1 is actually downregulated in response to SARS-CoV-2 infection. In fact, if you look at the underlying data points within the SARS-CoV-2 consensome, you'll see that ASAH1 is actually in the 97th percentile. So it is extremely sensitive to infection by SARS-CoV-2, and the effect of infection is to repress expression of this enzyme. And so what you can infer from that is that because SARS-CoV-2 is resulting in decreased levels of this enzyme, that would ultimately result in decreased levels of sphingosine 1 phosphate, and that may impact the ability of the lung epithelium to actually repair injury. So that's one hypothesis that you can use, using what's in the research literature and what's in SPP. You can build research hypotheses in this way. And so what we want to do in terms of the hypothesis center is to take a lot of what's right now, there's a lot of guesswork, for want of a better word, that goes into building hypotheses. And so what we want to do with the hypothesis center is to remove that, or not remove it, but reduce it as much as possible, so that researchers can make connections between or can use omics data sets to make connections between signaling nodes within their own specific area of interest. So with that, I will stop here, and with the last sort of 30 minutes or so that we have, I wanted to maybe get some insights